Hi guys, another Biology with Miss Cherry. This is the final part of the sort of genetics, genetic crosses, statistics side of this module. So we're on lesson 9 now of module 7. We're about halfway through. I'm going to start today by looking at this question. So cystic fibrosis is caused by recessive allele small f. A small population of Sudanese homozygotes all suffer from severe CF. What is the probability of sufferers in this population having a genotype f? What is the frequency of the dominant F allele? And what is the frequency of the recessive F allele? So pause and have a go at thinking about this. Hopefully it should be commonsensical, but if not, challenge yourself. Use that noggin. Might be quite early in the morning. Still stretch your brain. And then play again when you are ready. Okay. So hopefully it was not too tricky a question for you. Now it tells you in the question that cystic fibrosis is a recessive allele caused disease, which means that homozygotes that suffer from cystic fibrosis are going to have two small f's as their genotype. This means that there's a probability of 1 of the population being homozygous recessive. Now this means that in terms of frequency of the alleles, there is a 0% frequency of that dominant F allele. None of that population are going to have that dominant F. It's a recessive condition, which means that we've got 100% frequency of the recessive F allele, the small f. Okay, so notice the difference there. Probability is a decimal and frequency is a percent. Take that forward for the rest of the lesson. Now, if you weren't too sure about that, spend some time looking over the question again, thinking about what I've just been through. And if you're okay, let's get started. So today, the final lesson of this genetics side of things before we look at evolution and stuff later on is population genetics. And we're going to be looking at something called the Hardy-Weinberg principle. And this is a calculation used to have a look and calculate genotypic and phenotypic frequency, as well as the frequency of an allele arising in a population. So in the previous example, in that starter question, we did start to look at this, but there's a little bit more to it than that. So when you're ready, let's get started. So to begin with, we need to be aware of the key words that we're going to use throughout this lesson, and they are as follows. We've got population, gene pool, and allelic frequency. Okay, make sure we're confident in using those words. Could you have a go at thinking about what the blanks are? You might want to try writing them down and figure out, using your knowledge. You might want to do some research and find them out, but pause now, spend a second looking at these, thinking about what you think each of those words means, and play again when you're ready. So to begin with, we've got the word population, and you've probably heard population quite a lot, especially if you do something like geography. But population is a group of individuals of the same species, and that's the key thing. Otherwise, we're looking at like an ecosystem and things like that. So a group of individuals of the same species that occupies a particular space, so an environment, at a particular time, that are able to interbreed. And if they're the same species, we know that they should produce fertile offspring. And then we've got the gene pool. All of the alleles of all of the genes of all of the individuals in a population at a given time. So if we think about the class, that would be my alleles and your alleles and everyone else in the room's alleles, all put in this big mixing pot for all of the characteristics that we've got. So that I would have blue eye alleles into that pot, that gene pool. Some of you would have green eyes alleles put into that gene pool, and all of that stuff. And then we can relate the gene pool to allelic frequency. And allelic frequency is the amount of times that an allele occurs in a gene pool. So if there are lots of us with blue eyes in the class, then there's going to be a higher allelic frequency in the gene pool for 13D than there would be for maybe red eyes, because no one's got red eyes. Yeah, so we're linking these words together. So population links to gene pool, because we're looking at the alleles of the genes of the population at a given time, and then gene pool links to allelic frequency, because we're looking at the frequency of alleles in that gene pool for that population at that time. So let's take a look at this example. We've got the populations going to the gene pool. Now, cystic fibrosis is a recessive condition, and we said about this in the starter. And if we've got a recessive F allele, that starts to cause thick, sticky mucus that lines the trachea and the respiratory passages. And that's going to end up causing a kind of distress situation. We can't get enough oxygen, we become a little bit hypoxic, which is typical of cystic fibrosis. Okay? Now imagine we have got 15,000 people in a population. How many alleles of either F dominant or F recessive are there, and why? Pause, have a think about that question and play again when you're ready. So there's going to be 30,000 alleles in the gene pool of this population for that gene, okay, the big F or the little f. Each individual, remember, carries 2 alleles. So to get from population to gene pool, we have to look at the number of genes. So in this case, there's only 1. And then the number of alleles for each gene. And again, if we're thinking about humans, there are only 2. So number of genes times the number of alleles, and that gives us our gene pool number. Okay, common sense, standard maths, but we just need to clarify and maybe go over that again just to make sure. Before we go further in this lesson, we're going to have a look over this probability and frequency stuff again. So probabilities should be between 0 and 1. We said this at the beginning. You can have decimals, that is fine. And in terms of probability, 1 is absolutely certain. It's 100% going to happen. There's no chance that it won't happen. Whereas 0 is impossible. It's definitely not, under any circumstances, going to happen. And it's difficult to say in biology that there's absolutely 0 chance of something happening. So be very, very, very wary about that. Frequencies, however, should be written as percentages. So this allele appears maybe 67% of the time in this population gene pool. Okay, so let's have a look at how these can be used in action alongside some new equations. So bear with me, it's going to be a little bit complicated before it gets easier, but we'll get there. Now we're going to start looking at something called a Hardy-Weinberg equation. And this is used to calculate the frequency in percentage of alleles of a particular gene in a population. And there's a few caveats that are much like when we did the chi-squared test. There's a few premises that we have to meet before we can even consider using this equation, and this is the same situation. So the Hardy-Weinberg equation assumes that the proportion of dominant and recessive alleles in any gene in any population is the same between generations. So that implies that there's been no mutations, that there's been no movement of organisms in and out of our population, that natural selection hasn't taken place, so there's no advantageous alleles or anything like that. That the population size is large, so we've got a bigger sample size. And finally, mating within the population is random. We don't need to have populations that have got exclusive mating lines, for example. Things like zoos, for example. We can't use the Hardy-Weinberg equation if we've got controlled mating procedures. So let's have a look at why each of those are important, just in a little bit more detail, and then we'll actually get into the equation. And as we've said, we've got 5 different caveats that we've got to meet before we can even think about using the Hardy-Weinberg equation. And these are those. So no mutations means that we're going to get no new alleles arising. Yeah, they're not going to be able to have this new advantageous allele being mutated in. Population is isolated, so we're going to get no influx or efflux of alleles. There's going to be no migration into or out of that population either. Again, no natural selection. There's no advantageous alleles, so all alleles are equally likely to be inherited. The population is large, so it gives a big sample size. We're going to assume that that's standard if we've got a big sample size, that there's no other weird things going on. And finally, we've got that random mating, so there's no like alpha male or queen bee or something like that who is more likely to pass on their genes over the population. Okay, so all of these 5 things are important to bear in mind when we're working out whether we can actually apply the Hardy-Weinberg equation responsibly. And in some cases in an exam they might say, well, why can't we use the Hardy-Weinberg equation? and you would need to say something like the population isn't isolated, or that the mating isn't random, or there's potential for mutations to arise if this isn't met. Now, the Hardy-Weinberg equation also relies on the fact that our traits that we're using to observe, or we're observing, are monogenic. Okay, and for this, we're just going to give them the arbitrary letters capital A and lowercase a. And then we're going to take those capital A's and lowercase a's and we're going to change the letters for them. Okay, so my big A, my dominant allele, is now represented in terms of its frequency by the letter P. And likewise, my recessive lowercase a allele, its frequency is now represented by the letter Q. And for the Hardy-Weinberg equation, we have got two separate equations that interlink. The first one is P squared plus 2PQ plus Q squared equals 1. We'll go through what these mean in a second. And the second equation is P plus Q equals 1. You can see both of them are using the frequencies of the dominant and the recessive allele in their equations, and both of them equal to 1. Remember that 1 is a probability, and that it is definitely going to happen. Okay, so what we can see actually here is that P squared, if P is A, P squared is 2 A's. So it's our homozygous dominant genotype. Q squared is therefore our homozygous recessive genotype. And finally, PQ is either, or 2PQ should I say, is either big A, little a, or little a, big A. And those are our heterozygous genotype. Okay, now we're going to take a look at what these do in an exam question situation, which might make it a little bit more sensical for you. But if we take this information down here and we bung it into question 1, it should hopefully make a little bit, or equation 1, sorry, it should make a little bit more sense. So what equation 1 is actually saying is that the frequency of a homozygous dominant genotype plus the two heterozygous genotypes, okay, so either big A, little a, and little a, big A, and the frequency of the homozygous recessive genotype equals 1. And that makes sense. Yeah, there's going to be no random mutations that mean that someone's going to bring a different allele in, because this is one of those caveats, the important things that keep this working. Okay, and then if we think about the second equation, the frequency of the dominant allele in the whole population, so from both the homozygous dominant and the heterozygous genotypes, plus the frequency of the recessive, and let me change that back to black because that makes a little bit more sense, then the frequency of the recessive A allele, again, from either the homozygous recessive or the heterozygous genotypes, is also going to equal 1. It's just commonsensical. We can't get any other alleles in that population because that's one of the things that we've said. No changes, no variations, and we're working with these two equations in the middle for the rest of the lesson. So let's have a look at this exam question, and we need to think about what are our important bits of information from this question. So spend some time reading. I'm going to dictate it to you as well. The Hardy-Weinberg principle, represented by the equations below, can be used to estimate the frequency of alleles in a population. Albino rabbits have white fur as these individuals are unable to produce the pigment melanin. The ability to produce melanin is controlled by a gene with a dominant allele B, or big B, resulting in brown fur, and a recessive allele little b, resulting in an albino. Of the 60 rabbits in a pet shop, 45 are brown. Now, looking at the information here, I've got a few key points. I know that I'm going to be using these equations. It tells me that melanin, or the colour pigment, is controlled by a dominant allele, and that gives me brown fur. Recessive allele little b gives me an albino fur, or a white or pigmentless fur. And of 60 rabbits in a pet shop, so that's my sample size, 45 of them are brown. Now we've got to do the Hardy-Weinberg equation to calculate the frequency of the dominant allele in this group. And we're going to take a look at how we can do this on the next slide. Now the first thing I'm going to do is I'm going to find out what proportion of my population are recessive. Because we know that for recessive phenotypes to manifest themselves, we need to be homozygous recessive. So if 45 out of 60 are brown, that means that 15 out of 60 of our population are albino. Okay? And those albinos are going to have the genotype little b, little b. And remember, little b is the same as q squared. Okay, so if you wanted to, in your exam, when you get a question like this and they've given you the alleles to use, you can write them at the top. So you can see here that I'm changing what p squared means, and then what q squared means, and what the 2pq means as well, just to make it a little bit easier in my head. Now I know that 15 out of 60 is the same as q squared, and that means that q squared is the same as 0.25. And therefore, if we square root both sides, that gives me a value for q, which is 0.5. Okay, now as we can see from this example, the question was asking us for the frequency of the dominant allele, but we can't go straight in with the 45 out of 60 rabbits being brown because we don't know if they're heterozygous, in which case they've still got that recessive A allele, or whether they're homozygous. So as a rule of thumb, for all of these questions, always start by calculating the q squared and then the q value first. Okay, so we're not done yet, but we're working off of this calculated q value, the frequency or the probability of the recessive A allele, the lowercase a, appearing in our population. So stick with me, we're nearly there. Now we know that our q value is 0.5, and we're going to apply that to our equation, p plus q equals 1, because all we're doing is looking for the probability of the dominant allele, okay, the frequency of the dominant allele. So p plus q equals 1, and we've got a q value, that recessive A allele, of 0.5, which gives us a p value of 0.5 as well. Okay, so you substitute in, you move some stuff around, and you make p the subject. So the probability of the dominant allele in that population is 0.5, or it has a frequency of 50%. And that's that question done. Not too difficult, just a little bit of lateral thinking. So now it's your turn. Mucopolysaccharidosis is caused by a recessive allele little a. It affects 1 in 25,000 people. What is the incidence of carriers for MPS? I want you to think about how we'll tackle this and think about what you know. I'm going to write the equations at the side for you. So p squared plus 2pq plus q squared equals 1, and p plus q equals 1 as well. Think about what it means to be a carrier. Think about what it means for a recessive allele to cause a disease. Look at the incidence of it. Okay, and we're looking for those carriers. So you can pause, maybe spend 5 minutes having a go, and then I'm going to show you how to do it on the next slide. So pause now, play again when you're ready. So to begin with, we know that MPS is caused by 2 recessive A alleles. It's a recessive condition, we need both of them. Okay, carriers are going to be heterozygotes. Okay, so there's our big A, or little a, big A. And for both of those, the 2 little a's are actually coded for in the equations by q squared, and the carriers are our 2 PQs. Okay? And we know that there are 1 in 25,000 people with this condition. So 1 in 25,000 is the same value for Q squared. We square root that, it gives us a Q value of 0.0063. Okay, so we just write that in there. Okay, now remember, I'm looking for the carriers, and those are my 2pq. So what I can do now is I can use my Q value and put it in the P plus Q equals 1 equation. So P plus 0.0063 equals 1, and therefore P is equal to 0.9937. Okay, so now I've got my P value, which is 0.9937, and my Q value, which is 0.0063. And the final thing to do is find out what my carriers are. Okay, now we know that it's 2pq, so all I'm going to do is 2pq is equal to 2 times my P value, 0.9937, times my Q value, 0.0063, smash that in a calculator, and it comes out as 0.0125. Okay, that's the same as a frequency of 1.25%. Okay, so you can see there that I've used, and I'm going to highlight these in separate colours for you, I've used the information from the question to work out Q. I've then taken that Q value and I've used it to work out P, and then I've taken that P and Q value and applied it to my final equation that we've used not so far, so that 2pq, and given myself a frequency value, 1.25%. Okay, if you're not too sure about how to do this, let me know, pop up on chat, ask the questions, and then hopefully you should be able to have a go at the examples on the next few slides. Now it's your turn again. We've got 1,000 fruit flies and 640 of them have red eyes, while the remainder have sepia eyes. And sepia eyes are recessive to red eyes. And now how many individuals in that population would you expect to be homozygous for the red eye colour? And again, I'm going to give you the two equations just so you can see them being written, and you know that that's the first thing that you need to do for every equation that has the Hardy-Weinberg equation involved. Okay, so P squared plus 2pq plus Q squared equals 1, and likewise P plus Q equals 1 as well. So pause, spend about 5 minutes doing this, show your workings out clearly. There's no point waffling around your page getting 1,000 different numbers everywhere. Show it as a nice flow chart. Start with one step, then move on to the next step. Use arrows if you need to show links between the ideas. Okay, pause, spend about 6 minutes on this and play again when you're ready. So answers wise, we've got lots of information here. We know that sepia eyes are recessive to red, and therefore those flies with sepia eyes, I'm just going to use E's for this. So I'm deciding to use the letters based on the trait I'm looking at. So sepia I know is going to be 2 lowercase e's or Q squared. All right. And in the question, it tells us that 640 of the 1,000 fruit flies have got red eyes, which means that everything else is going to be sepia-eyed. So 1,000 minus 640 is 360. So 360 out of 1,000 is going to be Q squared, which is the same as 0.36. Q squared, and then we square root that, which gives us a Q value of 0.6. And now that we know that our Q value is 0.6, we can start using our equations. So we're going to start using the P plus Q one. P plus Q is 1. P plus 0.6, because we're taking that from the calculation that we've just done. And therefore, P is equal to 0.4. All right. Now the final thing I'm doing is I'm looking for the organisms that are homozygous for the red eye colour. So I know that that means I'm looking for P squared. Okay, because remember that's going to be my two big E's, homozygous dominant for that red eye colour. And so all I do is I do my p-value times itself. So 0.4 times 0.4, which is 0.16. Now this is not quite the end, okay, because the question was quite explicit in that it said how many individuals would you expect to be homozygous for red eye colour. So if I know that 0.16 of the population are going to be homozygous dominant for that red eye colour, and my population is 1,000 flies big, then all I do is 0.16 times 1,000, and 160 flies are going to have that red eye colour. Okay, so there's lots of marks here that are available. I'm going to highlight where they would be. So the first one is to recognise that 360 flies would have the sepia eyes. The second mark is for Q, the third mark is for P, the fourth mark is for finding out what P squared is, and the fifth mark is for the number of flies. So there's a high chance if you didn't check the question properly, you would have just said that there's a 0.16 probability or a 16% chance, but that's not what it asked. It looked for how many individuals. So make sure you're reading the question really carefully to find out what it needs from you in your response. One final example before you guys have a go by yourself. Phenylketonuria is an autosomal recessive metabolic disorder with a 1 in 10,000 incidence. Approximately what percentage of the population are heterozygous for the recessive PKU allele? I'm not going to give you anything for this one. There's 5 marks. Think about what we did last time. Think about what we've done in all of the examples. And I will go through the workings in a minute when you play again, I guess. But spend 6 minutes or so trying to figure this out for yourself for me. Okay, pause and play again when you're ready. So let's take a look at the information that we've got on this question. We know that it is a recessive metabolic disorder with a 1 in 10,000 incidence. And we're looking for the percent of the population that are heterozygous carriers for the recessive PKU allele. Now you can pick any letter you like to represent this condition, and I'm going to use a D for disease. So I know that someone with phenylketonuria, or however you say it, is going to have 2 little d's, and that's also going to be my Q squared. And we're looking for the heterozygous carriers, so big D, little d. And I know that those are going to be my 2PQ. Alright, so that's my base information. Now, much like the last question, we're going to take it in a few steps. So we're going to start at the very beginning. We're going to start by getting our Q squared values, and then we'll work through it like that. So I know that Q squared is 1 in 10,000 because that's my incidence. Alright, now for Q, I need to get the square root, and if you've got a calculator, you can just smash that in. Now I'm going to pause the video and type my answer into, or type my question into Google, and hope it churns out an answer there. And it did. It churned out an answer of 0.01. And now I can carry on exactly the same way as I did last time by using that to find my P. So P plus Q equals 1. P plus 0.01 equals 1. Oh, actually, that should be a plus. So now we're at P plus 0.01 equals 1, and therefore P is equal to 0.99. Okay, now that's another point that we're good to go. The final step is to get those heterozygous carriers, and we recognised at the very beginning that that's our 2PQ. Now we've got Q values, we've got P values, so all we've got to do is bung them into an equation. So 2 times 0.99 times 0.01 gives us 0.0198. And remember, it's asking for what percent of the population. So 0.0198 times 100 gives us 1.98%. Okay, so remember, you're just going to move the decimal point 2 places. And there we go. All done. So we're not going to do homework. We're in lockdown. You're at home. Every bit of work is homework. So On OneNote, you should see a file for the summary questions, and you should see a photo of the how science works, and you should see some photos of the practice questions A through to D from page 200. Now, normally at this point, I'd be getting a little bit antsy about the fact that we're very close to an exam, but we don't have an exam. So it's due by the end of the lesson. Can you please have a go at answering all of those questions to 3 significant figures? So remember, that's 3 values, or 4 if the first value is a 0, such as 0 point something something. Have a go at those. Can you write these out for me by hand, and then just put photos into OneNote? So pause, have a go, and then when you're done, play, and we'll do 1 exam question to finish off. So here we've got our final exam question. Black form ladybirds are recognised as dangerous by predators due to their coloration. Their dark colouring is caused by the presence of a dominant allele. Red form ladybirds do not carry this allele. The data in table 7.1 give the total numbers of the red form of ladybird found as 296 and the total number of black form ladybirds as 50. Use the Hardy-Weinberg principle and the figures above to calculate the frequency of the dominant allele P and the recessive allele Q in the 2-spot ladybird population. Show each step in your working and give your answers to 2 decimal places. 2 DP, so 2 numbers after the decimal point. And it's only a 3-mark answer, so it shouldn't be too long-winded, shouldn't be too complicated. But if you're not too sure, have a go. I need to see you having an attempt at it anyway, and then play again when you're ready to go through the answers, and we'll look at the mark scheme. So here are the answers. Should have recognised that Q squared is the red ladybirds. Oh, red. And that said that there were 296 out of the total number. So remember, there were 50 of the black ones and 296 of the red ones, which gives you a Q squared value of about 0.855. And therefore, Q, the square root of that answer, which should be about 0.92. Notice in the mark scheme that you can have an error carried forward for this question. Okay, so if you got the first part wrong, but you square rooted your previous answer, I'll highlight that at the top, then that's fine. You can give yourself that error carried forward mark. Okay, now the final answer, or the final part of the question, was for finding out what P is, and we know that P plus Q is 1, therefore 1 minus that 0.92, or the square root of Q squared, whatever you got for that value, gives you your answer, which should be about 0.08 to 2 decimal places. Okay, have a look down the right hand side. So this is the additional guidance section. This is where you can find out if you've got the correct answer. If P and Q are not given to 2 decimal places, then you get penalised 1 mark straight away. And you can see there's lots of guidance along the right hand side. So that means there's lots of areas where they're trying to give you marks, even if you haven't got the proper answer, like the correct full answer. This is why it's so important for these questions, especially all of the calculations, that you are laying them out in a nice, neat way. So even if you look at mine, although it's quite scruffy, I know that Q squared is red, my Q squared is this value, and therefore Q is that, and P is that. I've not got numbers jumping around all of the page. It's a very simple and easy to see flow. I hope you find that useful. If you need anything, please let me know. Send me an email or something. Otherwise, good luck revising, and I'll see you next time. Bye bye.